The late-breaking abstract I'm presenting here at ESMO is really exciting. We know for a long time that tumor metabolism is very interesting and potentially targetable with drugs. So the study I'm presenting here is a phase 1 dose escalation and dose expansion study in multiple tumor types of a new medication called TVB-2640. It's a tablet which is taken for 21 days as monotherapy and then 28 days in combination with chemotherapy. So the FASN pathway, it's very interesting and important. FASN, we know the fatty acid synthase is important and has been seen and demonstrated to be a mechanism of tumor resistance, multi-drug resistant, and is seen in several tumor types. So FASN produces palmitate. Palmitate is a very basic fatty acid and then gets complicated and more complex, built into tumor membranes, but also interacts with the canonical pathway, the Wnt pathway, and the PS3 kinase pathway. So a key role of palmitate in activating these pathways, and it seems to be that cancer is using and hijacking this lipid synthesis pathway. TVB-2640 is the first drug which really is targeting FASN, and the results are insofar interesting here that we defined a dose, and we defined the maximum tolerated dose at 100 milligrams per meter square as monotherapy and in combination with chemotherapy. We treated now in both mono and chemotherapy combination around 80 patients. And interesting, what we have seen is that the toxicity, the side effect profile is in line with our preclinical models. It's mainly about skin toxicity, hand-foot syndrome, and also eye toxicity in terms of uveitis and dry eyes. So other toxicities are very mild. The drug is generally very well tolerated, and these side effects I just described are very well manageable in a way. We have seen interesting very early signals of tumor efficacy in terms of disease stabilization. Several patients had disease stabilization for more than 12 weeks. Actually, in fact, we have also seen patients with confirmed partial response, and that is really interesting to follow up. So this study is currently ongoing in dose expansion, and obviously further results in terms of efficacy are awaited. Interesting in terms of biomarker analysis, we have also seen that the palmitate, that's the kind of end result, the target of FASN inhibition, is reduced in our patients in their blood serum. And also we could see in paired tumor biopsies that the phospho-AKT signaling is downregulated as well, in line with our preclinical models. So overall, very exciting, very early data, but we certainly can see that the biomarker analysis is supportive that we're actually hitting and manipulating the FASN inhibition, and also the early very efficacy results are encouraging, especially in the context that the drug is very well tolerated.